Welcome to our story about the fate of fall-applied anhydrous ammonia. This is Richard Mulvaney, and I'm with the Department of Natural Resources and Environmental Sciences, or NRES.
And I'm Mulvaney Griesheim, a graduate student in Dr. Mulvaney's lab. Today, we'll be talking about a trial conducted in the growing seasons of 17 and 18. We, along with our co-authors Tim Smith, Shelby Henning, and Alan Hertzberger, wanted to ask a fairly straightforward question, and that was, how efficient are fall applications of anhydrous ammonia, specifically in the Midwest?
Let's begin by comparing spring versus fall-applied N in Illinois. We do that here, and we find out, of course, that there have been ups and downs, which are mainly due to fall weather conditions. But over time, there's been a gradual increase in the proportion of fall-applied N. Now, anhydrous ammonia accounts for about 50% of the fertilizer N applied in this state, and about 50% of that anhydrous ammonia is applied in the fall.
There's a reason why producers like this type of application. First of all, it allows them to eliminate a task before the busy springtime with planting and other activities. There's also often improved soil conditions during this time for getting equipment in and out of the field. And there's also the improved infrastructure utilization. We have a spread throughout the year as opposed to using all of that equipment at one time. But despite all these benefits, there's one major downside, and that is that we're putting the fertilizer nitrogen out in the crop a good 6 months prior to crop and uptake. During this time, soil microbial activity can persist, and we can ultimately lose that nitrogen that we're putting out in the field.
A common way to deal with that downside is to combine the ammonia with N-Serve as a nitrification inhibitor. The purpose of the N-Serve is to inhibit the microbial process of nitrification that converts ammonium to nitrate. By slowing that conversion down, potentially we can reduce nitrate leaching and denitrification.
When evaluating this type of application, we wanted to think about it in terms of efficiency, specifically fertilizer nitrogen uptake efficiency, or FNUE. What this value is it's the percent of what we apply that gets into the crop. If we can choose application types that maximize efficiency, this is a win-win situation. We get most of the input into the crop, which is good for the producer, but that also means we're getting less of the input into the environment, which is also a great benefit. There's two different ways of measuring efficiency. First, the difference method, which is by far the most popular method for estimating efficiency. It's where we have fertilized and unfertilized plots, and we're looking at the difference in yield to estimate efficiency. But then there's the second way, and the way that we did it, which was to use N15. N15 is a stable heavy isotope. We can enrich a nitrogenous fertilizer source with this isotope and apply it just like a producer would, and then it acts as a tracer, so we can trace where the fertilizer nitrogen goes in the plant and soil system.
Unfortunately, there are some problems in working with N15 labeled ammonia. The first problem is simply the cost. So in using the rate that we used in this study, 200 pounds of N per acre, the price of 15 labeled ammonia would be a whopping $172,000 per acre. That's way beyond our budget. And so we had to work with small plots. But there's other problems that come up as well. And the next one we list is the need for care in dealing with the grinding, the analysis, and the N15 determination. We need to avoid cross-contamination of samples. And then we have to deal with the properties of anhydrous ammonia. It's a toxic liquefied gas. We're going to need to apply it to small plots because we can't afford large ones. And we're going to need to make those applications in a way that simulates what farmers do in the field.
So because this work has not been done before, there was a little bit of groundwork that needed to be done. And this is detailed in a paper, if you're interested, on the screen. The three components of this system that we needed to make, design, and build were, first, the manifold system to cryogenically transfer known amounts of labeled and unlabeled anhydrous ammonia into a collection tank. This collection tank's pictured towards the center of the screen. Not only could house the anhydrous ammonia, but allowed us to introduce a nitrification inhibitor by the sidearm. And this collection tank was also able to marry to the toolbar pictured on the right that was modified by Tim Smith and allowed us to be able to make those small plot applications in a way that was realistic or in keeping with how producers make their applications. Here's a short video of the application. So now that we have all the tools to make our application, we can move on to the details of the trial.
This slide summarizes how the work was done. It was done at 3 sites in each of 2 years, and those sites were on the same 3 soil series. The series were sable, which is a prairie soil, and it was in a corn-soybean rotation. We had a timber soil, Birkbeck, that was also in corn-soybean. And then third, we had another prairie soil, Elburn, under continuous corn. The plot layout is shown in the diagram on the right side of the slide, and our plots measured only 7.5 feet wide by 10 feet long. And in those plots, we're going to be evaluating 3 different treatments. One was the use of 200 pounds of N per acre. And the second was the same N rate, but with the addition of N-serve. And then thirdly, we had an untreated check with no N and no N-serve to help us estimate efficiency by the difference method. In all sites, there were 3 replicates of these 3 treatments.
So once these plots were established, we applied the labeled anhydrous ammonia, we allowed the crops to come to physiological maturity, and everything was hand harvested and separated into grain, leaves, stalks, and husks. These parts were then dried down and ground so that we could process them via Kjeldahl digestion and diffusion to determine the N content of these plant parts. The last step was carried out using a piece of equipment called a mass spectrometer. And what this piece of equipment does is it allows us to determine the N15 content of those samples. With this information, we can decide what % came from the soil and what % came from the labeled fertilizer that we had applied. So we have the total weight of all the plant parts, we have the N content, and finally we have the source of where that N came from.
Now, before we get into the data from this study, let's compare the 6 different sites where the work was done. And we're doing that here with a couple different properties. On the left, we have the potentially mineralizable soil N that was estimated by the Illinois Soil N test. That's telling us how much nitrogen the soil could supply. On the right-hand axis, we have % organic matter. And as we look at both of these properties, we notice that the 2 prairie soils, Sable and Elburn, were quite a bit higher than the 2 timber soils on Birkbeck. Now you'll notice that all of the sites differed in both properties. And again, that's because the work was done on different sites each year. Now notice the Birkbeck sites, they differed a lot more than the prairie sites. And in particular, we had a higher, more fertile soil in 2017 on that Birkbeck site and a much more depleted Birkbeck in 2018.
As we start looking at the data, we'll start with grain yield. We have our 6 sites here, each with 3 treatments. Minus NS would be our 200-pound application of labeled anhydrous without N-serve, and then plus NS would be the 200 pounds of N as anhydrous with N-serve. And then lastly, we'll have our UTC, which stands for untreated check. These are the plots where we had no addition of nitrogen or N-serve. Basically what the soil itself is providing in terms of fertility. The first comparison I'd like to make is between the UTC and our treatment plots. And what we'll be thinking about here is just the nitrogen. So did we see a significant effect when we added nitrogen to the plot? Was there an increase in yield with those 200-pound additions of nitrogen in the fall. We actually only had 4 sites where this was the case. This was site 3, 4, 5, and 6. Site 1 did not have a significant effect of the addition of N because it was so high in native fertility. What the crop needed was already present in the soil, so adding the additional nitrogen had no effect on yield. For the second site, it was a little bit different of a story. This site had previously been pastured. This means that we would have had a high spatial variability in N supplying power, and this is why we did not see effect there. The variation within the field fertility was too high. The last comparison I'll be making is between the minus NS and the plus NS treatment, looking and honing in on N-serve. Did we see a significant yield effect when we added N-serve? There was actually only one site where this was the case, and that was Elburn, site 6. At this site, you'll see there was a significant decrease in yield. So the only significant effect of N-serve on grain yield was a decrease at site 6.
Now we come to the main focus of the entire project. Estimating how much of that fall applied anhydrous ammonia ended up in the corn crop. We do it here, reporting the data in terms of FNUE as a percentage. We compare all 6 of the sites that we studied, and at each site we had both with and without the use of N-serve or NS. We show for each trial the uptake of the N15 in the grain, that's the red part of the bars, and then the additional height in the gray part that depicts the uptake in stover. For the grain efficiency, the numbers range from 12 to 34%, and they averaged 21%. For the total above ground efficiency, including both grain and stover, the range was from 16 to 42% with an average of 28%. So these numbers are telling us that the majority of the anhydrous ammonia applied in the fall does not end up in the corn. Now there were differences among the sites, and note in particular that the highest bars showed up for that Birkbeck site in 2018. I'll remind you that that was the poorest soil we studied. That soil had the lowest amount of N that it could supply, and so the corn crop took up more fertilizer.
The last measurement we'll be looking at is NDFF and NDFS. A good way to think about this is if we were to take all the nitrogen in the crop, it's what percent came from the fertilizer, so NDFF, and what percent came from the soil, NDFS. Here's the data. Again, we have both of our treatments without N-serve and with N-serve at our 6 sites. The red shows the percent of N in the crop that came from the soil, while the gray shows the percent of N in the crop that came from the fertilizer. The big picture story from this data is the fact that NDFS was always higher than NDFF. Another way to think about this is that the soil was always the dominant source of N for the crop. It didn't matter what year we were looking at, what treatment, what site, that was always true.
Now let's summarize the findings with these 3 points. The first reminds us that the uptake efficiency of the fertilizer N was only 21% of the grain on average and 28% for the total above ground biomass. So again, the majority of that ammonia does not end up in the corn. The second point of summary is to remind you of what Kelsey told you, that the only significant effect of N-serve on yield was a decrease that occurred for continuous corn in 2018. We saw no significant increases in yield due to N-serve. And then lastly, to remind you of the major point that Kelsey covered in the previous slide, that the soil was the main source of N for crop uptake. The fertilizer was the minor source.
We appreciate your time and attention and would welcome any questions or dialogue that you'd like to have with us Our contact information is at the bottom of the screen.